So let's talk first about the first group of tests, which are the phenotypic methods. Remember, with phenotypic, you're actually visually observing a change. So examples of phenotypic methods, first and easiest of all is just an immediate direct examination. First, we could just use a microscope. The most common and most important test of all when we're identifying a bacterial infection is the Gram stain, as you know. That is the first and foremost, gives you the most information that you're going to need. Is it a Gram positive or Gram negative bacteria? As you know, some bacteria do not Gram stain properly because they have a waxy cell wall. The Mycobacterium, for example. So for them, we would use acid-fast stain. So if you suspected a Mycobacterium infection, you would use acid-fast stain. Now, as you know, the Gram stain does give you a lot of information, whether the bacteria is Gram positive or Gram negative, but it doesn't tell you specifically what the pathogen is. For that, you can actually use something called a direct immunofluorescent antibody. So how this works is there's a known antibody that they sell commercially in a vial. And remember, antibodies are these Y-shaped proteins and they attach to the antigen. The antigen attaches at the arms of the antibody. And antibodies are very specific to their antigen. So they'll only attach to those specific antigen in most cases. Sometimes there are false negatives, of course. So for example, if you had something like syphilis, remember syphilis is a spirochete bacteria, and spirochetes do not Gram stain well. They're actually very difficult to see under the microscope. They're very, very difficult to stain, and there might not be very many of them in the sample. So what you can do is you can take this antibody, which will attach specifically to the syphilis, the treponema antigens, and then at the end of the antibody here, there's a fluorescent tag. And what you do is you expose this to ultraviolet light, and it actually allows the bacteria to fluoresce, such as in this picture. You see these orange and green corkscrew-shaped bacteria. So this could be done for something like treponema, or they do this a lot for Lyme disease. If they're looking for Borrelia, another spirochete, they can use this immunofluorescent testing. Another time we might use this direct microscopy examination is if we need to differentiate if the pathogen is either bacterial or fungal. A lot of times they'll do this for like skin infections. If somebody comes with a rash and they don't know if it's a yeast infection or a bacterial infection, they could simply take a swab or scraping of the rash. They could really quickly look at it under the microscope and say, hey, it's fungal, we need to use an antifungal skin treatment, or if it's bacterial, they can prescribe the proper treatment for it. Sometimes it's necessary to actually cultivate the bacteria on agar like we've done in our lab. And maybe that's because there's not enough bacteria in the sample, or for example, if we take a fecal sample and we need to characterize what type of bacteria is causing gastrointestinal issues, we'll often use the selective and differential media. So if we have to, we'll use isolation media. Most specifically, we'll use either a selective or a differential media. Or if we need to grow more of the organism in order to do further tests, we could use an enrichment media. Or if there's a lot of bacteria in the sample because there's a lot of normal flora, like if we took a fecal sample and we thought there was a pathogen there's a lot of normal flora there as well, we can use an enrichment media to try to enrich for the specific pathogen. And then the last phenotypic method is biochemical testing, which basically we're looking for the presence or absence of certain enzymes. Most commonly, we're looking for catalase, so that we can differentiate between staph or strep, for example. We might look for coagulase, which, as you know, is only produced by Staph aureus. We could do oxidase testing. There's a wide variety of testing we could do in order to really pinpoint the specific strain of pathogen. Now, as always, there's some certain bacteria that do not follow the rules. They're very difficult to work with in the lab. So we might have some miscellaneous or very specific tests used if we really need to pinpoint certain strains of bacteria or for those really stubborn or fastidious species. So for example, we might do something called phage typing. So phage are viruses that infect bacteria. Some bacteria naturally carry these phage genes inside of them. It's just part of their genetic makeup. So for example, different species of Salmonella, some more virulent species, may carry phage. So what we can do is we take a petri dish, an agar petri dish, we make a lawn of Salmonella all over the dish. So you'd make a lawn covering the plate. Then you would actually inoculate some phage. And phage kind of act like antibiotics in that you might see like a zone of clearing where the phage has killed off the Salmonella. So if you see the zone of clearing, that means this strain of Salmonella was sensitive to this phage. On the other hand, if the Salmonella still grows in the presence of the phage, it's resistant to it. So that's how you can differentiate different species of Salmonella. This is also done for different species of staph. As I mentioned, some bacteria we cannot even grow on agar. They just don't grow on agar. They only grow in live animals. So for example, Mycobacterium leprae, it only can grow in a live organism. And actually, we can grow it in an armadillo. So if we, for some reason, needed to test a strain of Mycobacterium leprae further, we could grow it in an animal in the lab. Same thing with Treponema pallidum, which causes syphilis. It does not grow in the lab on agar. We have to put it in animal species. Lastly, we might use antimicrobial sensitivity testing or Kirby-Bauer testing, which we're going to do in lab this week, where you put a lawn of bacteria on the dish and you put these little discs that contain different antibiotics, and you look to see if the bacteria still grows in the presence of the antibiotic or if you see that zone of clearing again. So not only can this be used to identify which drug would be the best treatment for the organism, but it also can be used to identify specific species or strains of some organisms. So we might use this, for example, for strep, Pseudomonas, and some species of Clostridium. Usually when we do phenotypic testing, we don't just do one test, we do all of the ones I just told you about. And there's these things called flowcharts, and you don't need to memorize these because most labs will have giant books of these flowcharts that you can go to and look at. So basically, for these flowcharts, you always will start with a gram stain, and you'll first look at the shape. So for example, you see a bacillus-shaped bacteria. Now, if this bacteria is purple, you know it's gram-positive. If it's pink, you know it's gram-negative. So, for example, if we found it was a gram-positive bacillus, next we would maybe conduct an endospore stain. If it does form endospores, we know it's either bacillus or Clostridium. From there, we know Clostridium are obligate anaerobes, whereas bacillus are facultative, so we could do oxygen testing. If the bacteria did not form spores, we then might conduct an acid-fast test. If it was acid-fast, we know it's probably Mycobacterium. So you get the picture. You can use these flowcharts to further identify usually the genus of bacteria that you're dealing with. If you want to look more specifically at the species or strain, we usually need to go on to the next type of test, the genotypic test.